Alex and I are going to do the wrap-up on the rest of today's session. We're going to narrow the focus a bit. We'll be talking about specifically Globus Genomics and use of that particular platform in our work with Nancy and her team over the last 1 to 2 years or so. I'll provide a bit of an overview on Globus Genomics as kind of a foundational piece. And then we had done some extensions on it, developed some very innovative pipelines through our collaboration with the Nancy's group, and Alex will go ahead and highlight some of those, some of the work we did, some of the results, and as well as some potential future directions. So kind of starting with what is Globus Genomics? Probably put simply, it's a next-gen sequencing analysis platform delivered as a software as a service. Researchers can define, create their own custom workflows using the integrated Galaxy platform capabilities. Researchers likewise can streamline their data movement and data sharing with the integrated Globus file transfer capabilities. And the whole thing is back-ended on Amazon Web Services, cloud-based, and kind of underscore a point Nancy had raised. In some cases, we have a lot of groups running on their local infrastructure and again, getting maybe select data into a cloud environment where there's much larger data sets and reference data and broader classes of data to work with. That seems to be kind of modality of a lot of the groups that we're working with are likewise going towards.
And I just wanted to make a comment. So my friends at UCLA, where they're doing a lot of big data and have to move a lot, rave about using how much easier it's been since they've gone to Globus.
Thank you. Yeah, thank you for that feedback. And the other point with back-ending it on Amazon Web Services, we found that a lot of groups, they can manage reasonably well local resources unless they have larger and larger scale of analysis they need to get done. And so we try to leverage the both scale-out and elastic properties of Amazon by provisioning resources on demand to run the workflows of interest. So just a bit more background on Globus Genomics. It's been developed over the last few years by a team at the Computation Institute. We've both developed and enhanced the platform, operated, provide support for it. And our chief goal in kind of doing it was to provide an advanced set of capabilities for researchers, and especially the biomedical space, to be able to get access to large sets of tools, get access to new sets of tools, evolving technologies, and kind of new and innovative algorithms, really as an aid to help them achieve their own scientific aims and further their research objectives. Globus Genomics is also part of a broader set of efforts going on and the kind of umbrella of Globus-related kind of development. And the broader initiative is to address even, I would say, a larger set of challenges associated with managing and working with research data. So just a little bit of motivation on why we started down the path of the sequencing analysis platform. It really started with a few researchers that we were working with and trying to help them with their kind of data movement, data sharing challenges. But we found in discussions with them that was really half the battle for them. Once they got the data even moved, and there's a lot of issues in kind of getting it from, let's say, point A to point B, is how best to get the analysis done, and especially at larger and larger scales. So there's two classes of problems that we are looking at, and I'll describe a little bit of the current challenges researchers are facing, and then talk a little bit about the solution we put together. So on the data movement side of things, researcher may be getting their sample sequenced at multiple facilities. It could be a facility on campus, across the country, overseas. So data is distributed, and the need to kind of get that data into their labs and ideally into an infrastructure where there's much larger reference and other data sets to be able to compare against is critically important. Most of the groups we were talking with pretty much relied on just getting hard disks shipped around via FedEx. In some ways, it doesn't seem like a overnight delivery is too big a deal, but a lot of sequencing centers, they kind of queue up the samples that needed to be analyzed or raw data to be generated against. They pack the results on a hard disk, and what could have been transferred when the first sample was done might be actually sent out instead of hours, weeks later to the researchers. And so just getting larger and larger sets of raw data sent around was a bit of a challenge. It's compounded when you want to share that data with other groups, other collaborators, and so shipping disks from a sequencing center to your lab is maybe a headache enough. Getting it then shipped to a bunch of collaborators where you have the data that you'd like to share, kind of just exacerbates the problem. So folks have typically turned to FTP, SCP, but when you're looking at gigs, tens of gigs, hundreds of gigs, and even terabytes of data, it becomes a performance nightmare. Extraordinarily hard to actually manage that type of transfer with kind of antiquated tools. And then even once the data does get to the destination, a lot of groups struggle with storage facilities which are at or pretty much near capacity. So even if a hard disk show up at a researcher's lab, we'd see them effectively getting piled up until they could finish analysis on their cluster, delete data that was already basically there, and make room for the new data to go ahead and mount and download. So you can imagine there's all manner of inefficiency in the current kind of state of things. So then even once the data does get moved over, kind of the second part of the challenge for the researchers was how best to get the data analyzed. We had groups that would either recruit or try to cultivate resident experts in not only scripting, but basically in kind of installing tools, managing tools to run in their environment. So when they go about actually defining a pipeline, they have to get the requisite set of tools identified. They have to get those installed and validated in their local environment. They want to be on the lookout for any new or interesting tools and basically go through that same cycle over and over again. And then once they did have the right set of tools set up and validated and working, they'd go ahead and turn to scripting to go ahead and stepwise kind of sequentially execute those tools to get their pipelines running. Maintaining these scripts, as one can imagine, could be very error-prone, gets to be messy. And the other issue is even if one cultivated that level of expertise in a group, which is really meant for biological research, not so much IT expertise or even scripting expertise. If that individual walks out the door, gets an opportunity, postdoc goes on to another institution, they pretty much walk out with the knowledge as they go, and the team is kind of left with, we're almost restarting from scratch how we can go about getting our analysis done. I guess one other point to raise here is, even with this type of an environment, it can be rather fragile, as I've described, and there's not necessarily a good way to maintain the histories of all the analysis that is done, which I think is increasingly important. So we thought there was certainly a better way in addressing this twofold set of challenges, and we kind of brought Globus into the picture for the data management, data sharing related issues. The notion here is to make use of Globus endpoints, and that is really to kind of set up facility at either sequencing center or multiple sequencing centers. Could be at the PI's desktop, laptop machine in their local cluster, and even better in their kind of cloud environment where scale-out analysis can be accommodated. And one can as well set up endpoints at collaborating institutions. So effectively, when you have larger sets of data that you want to get moved, by having these endpoints set up in this kind of ecosystem in place, it's a rather simple task of a drag and drop with an intuitive interface to move one or hundreds of thousands of files and small data sets up to terabytes of data. And so that's kind of the intent of getting the Globus transfer, Globus sharing capabilities kind of factored into the solution. What we did on top of that was integrate Globus with the open source Galaxy-based workflow system. Galaxy has been in wide use with the next-gen sequencing community. Provides a very intuitive interface to kind of drag and drop of creating your workflows. Has a good-sized workbench to draw from, a number of next-gen sequencing tools, from alignment, variant calling, QC tools, etc., and some even downstream analysis tools that are provided. And then one can easily modify existing pipelines or create new ones completely custom, and then be able to execute them. And as I mentioned earlier, we've pretty much back-ended it with Amazon with the goal of letting one be able to do analysis at fairly large scale. So even once the data is moved and once the pipeline is defined and validated, more and more of our groups that we're working with, they're not running on single data sets, a single exome, or even a whole genome. They're wanting to interrogate it against tens or hundreds of these data sets. Local and compute and storage just normally wouldn't allow for that. So you can effectively use the scale-up capability with Amazon, and then the elastic properties as well. And so, for example, we had a group out at the University of Washington. They've started off doing their own exomes. It would take them anywhere from 10 to 15 hours to run their pipeline on their local cluster. They're getting larger and larger data sets. They wanted to be able to run that at scale. Now they're running upwards of 30 or 50 exomes in parallel, roughly in the same time it took them to run the single one. And using the elastic properties of Amazon, the system basically creates a dynamic cluster on the fly. The analysis is done against all of the workflows, let's say in parallel, and then when the analysis is completed, all the resources are basically released, and so there's a fair bit of efficiency to be had. And that's in comparison to these local environments where one can either try to design for a peak load and then carry a lot of hardware costs when you're not running at peak, or what's worse for the researcher is design for what may be more of a steady state load and then rarely ever be able to hit kind of a peak demand. And I think with groups that we're working with, at least, they're seeing a lot more growth in both data sizes and number of samples that they're trying to get analysis done against. So I'll mention just a few ongoing efforts, and then I'll go ahead and turn it over to Alex, and he can detail a bit of the work we've done with Nancy's group using some of these capabilities. So we continue to add new tools to the workbench. The open source community in Galaxy does a fantastic job in having hundreds of tools available. What we have found is a lot of our collaborators, they're wanting, let's say, a new tool that they've heard about, added. They may not be able to wait for the n number of months before kind of the open source community kind of gets to wrapping it. So we go ahead and add these, validate them, do some performance optimizing so they run very efficiently within cloud resources. And so most of that is driven by collaborations and what our researchers kind of need us to add. We continue to investigate and implement new methods of performance optimization. The example I alluded to earlier where we do more of a parallel workflow execution where one can run on a single exome or 50 exomes at roughly the same time. We have that level already in place. We're doing some task level parallelism as well. And then Amazon, with new and kind of more advanced compute capabilities coming online, seems virtually every week or month. We experiment with new instance types, which are much higher memory, more cores, and then are able to tweak which tools can run optimally on that kind of changing, evolving set of technology to squeeze out as much performance and optimization as possible. So just a few examples of the type of work that we're doing. Running at larger and larger scale, we started off with exome and genomes on the order of single or double digit. We're finding groups wanting to run consistently larger and moving into the tens to hundreds to ideally eventually handling thousands with this type of modality. We will continue to incorporate efforts from the broader Globus team with extensions in and around research data management. That would be metadata definition and management, doing advanced querying, data publication services, and in some ways combining not just the raw genomic data, but outputs from the analysis from QC to variants that are identified, to as well other types of data where one can kind of look at the problem more holistically. Lastly, we continue to seek out and collaborate with several leading research groups. We that way kind of keep enhancing the platform, but make available state-of-the-art pipelines for broader use. And frankly, the work we did with Nancy and her team is probably one of the better illustrations of that. So with that, I'll go ahead and hand it off to Alex, and then maybe at the end we can deal with a few questions if there are any.
Good morning. So what I'm going to talk about is what we were able to do with in the collaboration with Nancy Cox and her group. And this collaboration started out back in the summer of 2012. And what they were working on at the time is an application of which is called the consensus genotyper for exome sequencing. What it basically did is you were able to run call variants on a number of data sets, a number of exome data sets, and then use this application to create a consensus of a rich quality set of variants. The challenges were that the data set was distributed at different locations, different points in different research groups had certain data sets, so they needed to be put together into a central place where we could use this complete data set to perform the different steps in the workflow. So we transferred, so another challenge was doing the transfer and sharing the data with the other researchers. And back then they were doing the data movement using SCP or just putting it in an FTP location. And so that didn't work well with the Globus Genomics integration. So we integrated some of the Globus transfer tools into, or we already had it in Globus Genomics, so we were able to use those tools to move the data efficiently into the centralized analysis location. The other shortcoming that they were having is that these data sets included, the one that they were working on was about 150, somewhere around there, 150 sample data sets, so 150 patient alignment files. And so performing that analysis on their machine would take several months to perform. So there were several steps that needed to be performed in order to get to the consensus part of the workflow. And so handling the complex analytical steps and all these tools in order to do the parallelization was not possible on their local server. So some of the goals that we had were to create the Globus Genomics instance for Nancy Cox and her group, such that the data that they have is not open to everybody, but only to the people that they want to share the data with, that have access to this instance. Also, we were able to transfer the data using the Globus data transfer tools by integrating Globus transfer tools into the Globus Genomics space. And basically make this Globus transfer tools as part of the workflow, such that the user doesn't have to worry first about manually moving the data using the transfer tool, but instead integrate it into the workflow such that the user just points it to where the data is located, and then as part of the general workflow, it just starts executing all the way to the end. We were able to integrate multiple variant calling tools one of the goals was to integrate variant calling tools so that it identifies SNPs and indels. So part of the consensus, the CGIS application is that it uses multiple variant colors. So and I'll show some of the variant colors that we had. So what you're trying to identify is what are the variations from the reference data set with respect to your data set, your samples, your patient samples. Right? And so in order to get a rich set, what they wanted to do is use multiple tools to call these variants, such as some of the tools that they were using was Freebase, Atlas, and some GATK tools, such as Unified Typer and Haplotype Color. So one of the other things about these tools is that in order to parallelize on a sample level or a chromosome level, it wasn't as streamlined as you would. It wasn't as streamlined as it should be. And it wasn't as nice for a researcher to come in and then have to, if your data set has 150 samples, it contains 150 samples, and on each one you have to call variants. If you have to manually, start the process for each one, then that's not a nice way to do it. So what we're able to do is also integrate into some of these tools Swift, so that in the back end it would automatically do all these parallelization for you. And so we're using this, we generated the workflows for the automated analysis, and then at the end, some of the goals for us also, once we had this consensus set of variants called, be able to share the data with other users, the workflows, and also the workflows such that if other users have their own data sets, they can start the analysis themselves as well. So for this collaboration with Nancy, the data was about over a terabyte, and this was transferred using the Globus data transfer tools. And it included 134 alignment files from the autism data set. And so, as I said, several variant calling tools were integrated into it, such as Atlas 2, GATK Unified Genotyper, and Haplotype Color, Freebase, and Samples and Pylon. And one of the things that we noticed is that you can increase the speed of it depending on the tool by parallelizing for calling the variants on a chromosomal basis. So for the human data sets, we were able to parallelize on multiple chromosomes. But then there were other tools like Atlas that you had to call it on a per sample basis. So you needed to parallelize it on 134 files. Okay, so again, we integrated Swift such that the user would only point to the location of where this data set, this compute data set is, and then in the back end it would take care of generating the necessary steps to submit the 134 parallel jobs. So the other thing that was done by Nancy Cox's group was the CGIS pipeline. So we're able to take her scripts, integrate it into the tool panel, so that once all these colors are done calling the variants for each of the tools, it could be fed into this CGIS tool and call a rich set of variants. So in general, or an overview of the CGIS workflow is the following. So you would take in the alignment files in BAM formats and then run it through the different variant colors. So we have GATK, the GATK workflow, the Freebase workflow, and the Atlas workflow, and each one is different. They have a number of different steps. They are not all the same. And then at the end, you have this variant calling file, variant call file, a VCF file, which you're going to feed into the CGIS pipeline, and then get the rich set of variants. So So the analysis was performed on multiple resources and the Amazon cloud. We used the worker nodes that we used were an N2 large machine which contained 8 CPUs and 64 gigs of RAM. And so we were able to do this for the 134 data sets altogether for each of these pipelines, which ran also in parallel separately. So the GATK pipeline took about 21 hours to complete for all the data sets, and it used about 240 node hours. Freebase took about 31 hours to complete. Atlas was the most compute intensive when it took 111 hours and over 600 node days. So that was the most expensive tool to run, because you couldn't run Atlas as a multi-sample in a multi-sample way. When you say you have to run it per sample and then basically use that individual column and create a multi-sample data set of Atlas. So that's why this tool took the most. But from the results that we were able to get is that Atlas was the one that was the most efficient in calling the variants, and the Nancy Cox group was most happy with the calls from the Atlas tool. After these 3 pipelines were complete, the individual pipelines were complete, then it ran the consensus caller, and it generated a high quality list of variants, and it was less than 0.01% of Mendel error rate for 3 variants. Okay, so now I'm showing you a little bit of how complicated some of these workflows look. So this is when we were trying to do the parallelization per chromosome for the data set. So you would run basically the same thing for each chromosome, the same freebase calling algorithm. But after we implemented the Swift into these tools for parallelization, since this freebase job is the one that's repeated over and over for the different chromosomes, this is the only place that the user would have to specify what data set, and then automatically in the back end it just gets fanned out, and then at the end it combines all the variants. So the goal is to simplify the analysis for the user, and we believe this simplified the analysis for the user significantly. And again, for the GATK unified phenotyper, the parallelization was also done on a per chromosome basis. So again, this step was the one that would fan out to 23 plus some other contigs that they wanted to check. And so this one we were able to simplify it using Swift, by integrating Swift. And this is what the Atlas genotyper tool looks like when you are modifying it through the Galaxy interface. So again, there's a lot of parallelization at this level, but through the integration that we are able to perform, it simplifies the analysis for the user. So at the end, the way to start a workflow is very simple. You fill out whether the input data sets, where they are located, and what location, or either their remote locations, or if they are already locally in this analysis server, then you would indicate where that is. And then you just hit run workflow after you build those different workflows that I showed you before. And then it starts submitting the jobs automatically. So some of the ongoing work and future direction. One is we are able to share this workflow with other groups. We already worked with several groups they're interested in using this. So we're able to, by integrating all these tools and making it available through the Globus Genomics platform, they're able to log in, submit the jobs, and just for them. Also, we're adding other variant colors. So the nice thing about the consensus genotyper is that it's not limited to just those 3 variant colors, but you can also add others as new colors come along, or as users that we work with are interested in a particular variant color, and we could add that into the pipeline or into the tool set, such that they can just easily add another branch. We're also, so the work with NASCARs, they focused on SNPs. In a group that we're working with, they're interested in indels. So we are adding that people looking at testing all these testing the results with real standard verified indels. One of the collaborations that's on the road is it includes a data set that has more than 10,000. It's an autism data, another autism data set. It's in the cloud, so it would be easy to integrate it or move the data here. And it includes over 10,000 data sets. So right now we're not able to perform this type of analysis of just doing more than 10,000 in parallel. So that's something that we're working on and testing how to make it possible. Also, variant annotation. So right now we've only been doing the calling the cross-matching tools to do the variant annotation, such as animal right now. That's what we're using. So with that, I close and I just want to acknowledge NASCAR lab, Leah and Dasa have put in a lot of time working with us, and from our team, Ravi, Dina, and Lukash, and Ian. Thank you. Thank you guys.